Lovely. Thanks, Mariana. That's fantastic. So I guess what I thought I'd do just to round off this session a little bit. So my name's Jack Rogers. I'm based at the University of Birmingham. Is really just to sort of present some work that's used some of the analytical approaches that Mariana sort of alluded to as part of obviously the work we're doing in Psy-ShareD. Also some work that's been done as part of some of the Psy-ShareD data, so some of the data that's actually gone into the Psy-ShareD database, and also perhaps to give some people some ideas that might mean they want to sort of get involved, I suppose, with the Psy-ShareD database. So this is some work that was conducted by Dr. Paris Elusis. So Paris was a PhD student with us here in Birmingham and is now a lecturer in AI and mental health at King's College London. And this was work done as part of the ProNEA consortium. So what Paris did was to as we've heard from Mariana, use a sort of a series of support vector machines. So use machine learning models that are trained here from the ProNEA database on clinical symptoms, as well as looking at brain structure to try and really explore heterogeneity within the sample, look at comorbidity, factors of onset, duration of illness, and prognosis in different psychiatric disorders. So really to look at sort of shared, I suppose, and distinct illness features at the phenotypic level, particularly across clinical and neurocognitive performance, which is obviously what we have as part of the Psy-ShareD database, as well as looking at differences in brain structure, so grey matter volume. And so what Paris did was to generate these prototypic models here of recent onset psychosis. So this is what the ROP stands for, and recent onset depression. And train these different support vector machines using a toolbox called Neurominer, which people might be familiar with, to really try and separate based on these different data. So use a data-driven approach to try and separate patients with recent onset psychosis from recent onset depression. And as Mariana alluded to, so this was using various sort of iterative testing, cross-validation approaches, looking at the area under the curve, sensitivity, accuracy, and balanced accuracy processes. And what you can see here is that the model does quite a good job. So you can see the blue dots here are those people with with recent onset psychosis, and the red are those with recent onset depression, and the few cases where those were misclassified. And this is based on the clinical and neurocognitive data, as well as when we did the modeling based on, well, Paris did the modeling based on the brain structure. Taking this one step forward, what was also explored is looking at things like comorbidity. And what was interesting, and something that obviously we also might be interested in within the representative Psy-ShareD database, is thinking about perhaps the contribution of things like comorbidity, bipolar disorder, as was referred to earlier, things like trauma that has been mentioned as well. And what was interesting in this particular database as part of the ProNEA data is that there was a shift of comorbid patients, particularly those that were comorbid with recent onset psychosis and depression, towards more like the depression prototype. And this was observed at the clinical level, so within the models, and also not quite as to the same degree within the neurobiological model. So basically what this showed, at least to us, was that so that the sort of the pure recent onset psychosis group might have a sort of different, I suppose, phenotype to those with psychosis and comorbid depression, who show more sort of clinical and neurocognitive features to the recent onset depression with psychosis group, as well as the pure recent onset depression group. That was shown also within the brain structure. So what we were able to do with this approach is to look at the cross-validation ratio as well across the brain, looking at areas of reduced grey matter volume as well as increased grey matter volume across those that have a sort of, I suppose, more pure form of things like psychosis and depression, as well as exploring differences in brain structure, so overlap and distinct features of neurobiology in those that have different levels of comorbidity. And obviously this is something that we're very interested to do in a sample, a much bigger sample, as is the case with the Psy-ShareD database. The other thing we're really interested in, and this was alluded to a bit as well by Mariana in terms of when people were developing things like the harmonization protocols, and this is certainly true, I think, in sort of the next phase, I suppose, of the work for Psy-ShareD, for the Psy-ShareD data, is maybe thinking about the role of other biological features and how they might contribute to brain structure. So this includes potentially things like proteomics, transcriptomics, genomics, and something that we're really interested in is the role of inflammation in the development of different severe mental health disorders and the impact that has on the brain. So there's now a growing body of evidence, including some work from ourselves, showing that inflammation or changes in the immune system play a key role in the development of schizophrenia. And as we've heard today, obviously patients with schizophrenia and different forms of psychosis show a range of differences in brain structure, and as well as things like disordered synaptic pruning and other measures of change within the brain. But obviously we're trying to get at the root cause of what's going on there. And one candidate that we're particularly interested in are things like pro and anti-inflammatory markers in the blood, particularly things that are able to permeate the blood-brain barrier. So these include things like inflammatory cytokines like interleukin-6, interleukin receptors, as well as things like tumor necrosis factor alpha and other markers and proteins within the central nervous system that have been shown to contribute to increased risk for psychosis. The reason we're interested in things like R6 is primarily because of its permeability, so its ability to cross the blood-brain barrier and sort of draw in other inflammatory markers that might contribute to things like treatment resistance, as well as other clinical and cognitive outcomes. So as Simon referred to earlier, so thinking about sort of what features are driving differences in cognitive deficits within these different populations. However, to date, there's relatively few studies that have done that, particularly in the brain. And there's even fewer studies that have looked at things like how we might subtype different populations of psychosis based on immune active or immune inactive subgroups. So some very recent work that we've been doing, so this is work that was led by Alex Murray, who was a PhD student in our group, who's recently qualified, was to use again a sort of a machine learning approach, this time using a tool called HYDRA, which stands for Heterogeneity Through Discriminative Analysis. And what HYDRA essentially does is to, a bit like Marianne was alluding to in terms of the support vector machine approaches, is to try and separate the patient samples from the healthy controls, in this case by forming what's called a complex polytope classifier, which essentially tries to disentangle heterogeneity within the data, particularly for the clinical versus the control samples, by applying an adaptive form of k-means clustering and single linear and non-linear subclassifiers. And so this is, again, a potential approach that we could use in the context of, obviously, our large Psy-ShareD database. In this particular example, this was using data from the Benamin data set, so the benefit for minocycline data set, which was conducted with calibrators from Nottingham and from Manchester. And in this case, we've looked at whether we could subtype or subgroup people based on different inflammatory markers. So we're looking at things like interleukin-6, as I mentioned, as well as other pro and inflammatory cytokines, things like C-reactive protein, interferon gamma, and tumor necrosis factor alpha. And these are markers that have been already linked to changes in brain structure in psychosis, and have also been linked to different cognitive and clinical outcomes. So what we found, and this work is sort of currently under review, is interestingly, we could identify different inflammatory subgroups within our population. And so interestingly, what we found is that there are different profiles, I suppose, of grey matter volume reductions across these different subtypes of inflammatory profiles. So this is just really to highlight that actually what's quite nice is that we could use this approach to try and look at potential risk factors, I suppose, or even inform more targeted treatment approaches in different subgroup populations. So here we found, and this has been shown in depression as well, is that people with atypical or more altered C-reactive protein showed the most widespread reductions in the brain, and this included superior frontal, medial frontal parts of the brain, and they also showed the greatest cognitive deficits. And this included things like full-scale IQ testing, particularly subscale, so digit span testing, reading, and this was also mentioned by Mariana in terms of the sorts of metrics that we have as part of Psy-ShareD. The interleukin-6 cluster showed higher scores on PANS, so the positive and negative symptom scale, which is a clinical measure, particularly on the anxiodepressive dimension of PANS, and also showed overlapping but also distinct grey matter volume reductions within left superior and inferior temporal regions, so regions that have been implicated previously in psychosis. Interestingly, we also saw differences in what we call a non-inflamed cluster, so reductions in areas of the brain in people that didn't show sort of increased inflammation within these different inflammatory cytokines. And that's also interesting because obviously that suggests that these people might respond perhaps to more traditional methods, so more traditional antipsychotics or more traditional forms of treatment. The final thing I want to mention, and again, something that potentially could be explored using this sort of rich Psy-ShareD data, are things like Mendelian randomization. So thinking about predictive components, looking at that might predict changes in brain structure. So this was work conducted by a postdoc, Jackie Williams, who's now back in the States. This was work done using UK Biobank, but actually I think some of these ideas could also be applied to the Psy-ShareD database, which is really exciting. So particularly this work was looking at trying to explore the relationship between inflammation and changes in brain structure in vivo using genetically predicted markers of change. So it was hypothesized that genetically predicted levels of interleukin-6, interleukin-6 receptor activity would be associated with more grey matter volume increases or potentially decreases in the middle temporal gyrus, inferior temporal fusiform and frontal cortex, and also explore changes in things like cortical thickness. So obviously what we can also explore within the context of the Psy-ShareD data are things like surface-based morphometry, cortical thickness, etc. What was interesting is that interleukin-6 particularly predicted changes within these sort of middle temporal regions, but there were no significant associations for some of our other factors, including, interestingly in this case, no change for CRP, so C-reactive protein. So the strongest associations were with changes in grey matter volume, particularly sort of negative, so grey matter reductions associated with, in this case, our inflammatory response. So here it was sort of being driven by one particular marker. So that just gives, I think, sort of, that's just a quick snapshot, I suppose, of some of the work that's been ongoing that might give people some ideas about things that they might want to explore within the context of the Psy-ShareD data. So I'll stop sharing and obviously happy to answer any questions and pass back to the wider team. Thanks, everyone.
Thank you, Jack, for that final session. And I'd just like to thank everyone. I don't know how anyone can hear me. I'm coming closer. I'm coming closer to the microphone. Yeah, I'd just like to thank everyone who joined this afternoon. I'd like to thank Veena and Simon for the sessions earlier, and Lucia for joining and giving a rather unique perspective. And the session this afternoon, I think has been very helpful, and thank Mariana, Matthew, Jack, who's just spoke, Sarah and Ruba for all their contributions this afternoon. I suppose just to end, I'm hoping you can see the screen so that you'll see there's three links on there. So the first two are to papers that we have published, one in schizophrenia, the Nature Schizophrenia Journal. That's the short commentary piece that Simon and I have published. I think it just sort of outlines what we feel are some of the needs for Psy-ShareD. There's a preprint, a more detailed paper on the Medarvix preprint server, and that obviously is currently in peer review. It's in revision for human brain mapping, and we're hoping that'll be published early in 2025. If you want to look at the preprint, that's link should take you straight to it. And sorry. And we're going to put the links into the chat as well, so you should be able to log back onto Teams later, and they should all still be there. That's if I understand how it works correctly. And the final one is a link to a feedback form. It would be really useful just to get some feedback on today's session, what worked, what didn't work, what would be helpful maybe in future workshop sessions. That's kind of really what we want to find out. So, you know, I know it's a bit of a sort of, can be a bit of a pain and bit of extra time, but you know, if you can just sort of go on, and it shouldn't take you more than a few minutes just to click a few tick a few boxes and leave a couple of sentences of feedback, that'd be great. So I think I don't know, did we get any more questions coming in since Jack's finished his talk? So I think we want to sort of bring it to a conclusion now, but just before I do, are there any more questions, any hands up, or before we finish, anything coming through? Okay, I can count to 3. 1, 2, 3. Okay. Well, look, you know where we are if you need to contact us. Just one more time, just to say thank you for everyone who's joined today, and wherever you're joining from, have a good evening or rest of the day. Thank you very much for your time.